Oh, phonetically, it's exactly as it is spelled. So it's Nara Parameswaran.
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 Parameswaran. Okay, I'm sorry. Okay, all right. Okay.
So with that, Michael, well, it's 7 o'clock as the clock ticks. So it's as one of the things being an old program manager, one of the things I believe is punctuality. And people, as they join on, will have to play a little bit of catch up. And we are recording this, and this will be posted on the Scleroderma website, the Michigan chapter website, in the next few days. So if you miss something is missed, and we'll restate this at the end so people can watch what they need. So with no more further ado, Michael, I will let you take over, and I will just, when you need to index the slides, Doctor, just say next slide, and I will page to the next slide for you.
Will do. Thank you.
Thank you.
Good evening. I want to welcome everyone to the Scleroderma Foundation Michigan Chapter's web-based 2019 educational series. This webinar is being offered as part of our strategic planning efforts to move toward web-based learning experiences for the next 4 years in an effort to reach more patients, caregivers, and family members who are seeking to obtain accurate information about scleroderma. There are individuals from all over the world participating on this webinar, emphasizing that we can be connected and sit in front of our computers while staying in homes and our offices. This is the third webinar in the series focused on microbiome and how it affects your disease. There will be 3 more webinars presented in 2019, and you can the information can be located about them on Michigan Chapter's website. If you'd like more information about these, you can find the information on the National Weekly e-blast or on the Michigan Chapter's website, which is, all small letters, scleroderma.org/Michigan, and Michigan is spelled out. This evening, we have with us as a presenter, Dr. Nara Parameswaran. Dr. Parameswaran is currently a professor in the Department of Physiology at Michigan State University. He is also the associate chair from the Department of Physiology in Michigan State University, and he's also the undergraduate program director for physiology. He received his DMB, veterinary medicine, in 1994. In 1995, he received his MS in veterinary physiology and pharmacology. In 1999, he received his Ph.D. in physiology at Michigan State University, and he continues teaching and helping us to present tonight. And he is also numerous honors and awards, and he is co-author and editor of 2 books. One is The Potential of Probiotics as a Therapy for Osteoporosis, and Understanding the Gut-Bone Signaling Axis. Without further ado, Doctor.
Thank you. Thank you so much for the kind introduction. Hello, everyone. So what I'm going to do today is give you an overview of microbiome and how the microbiome affects various disease processes, and in part also explain a little bit about how it might be influencing autoimmune diseases. Next slide, please. So the number one thing that I would like to do is actually describe what microbiota is and what microbiome is, because these 2 terminologies are used interchangeably by a lot of people. And so let me first explain that and then give you an overall idea of what I'm going to do today in the next 1 hour. Next slide, please. So when we say microbiota, we are talking about a group of microorganisms that are living in a particular place, and we call that as a particular niche. And so when they live in the GI tract, we call them as gut microbiota. When they are on the skin, we call them as a skin microbiota. When they're in the lung, we call them as lung microbiota. So depending on where they are, we call them in relation to the organ. But these are the actual microorganisms that are there. These could be bacteria, these could be viruses, and so forth. And many of them are beneficial to our body. Next slide, please. When we say microbiome, what we're talking about is the genes that are expressed by these given microbiota. So each microbe, bacteria or viruses, express specific set of genes, and those genes have certain functions. And so when we say microbiome, we're talking about the genes that are being expressed by these group of microbes in the body. Next slide, please. So what I'm going to do in terms of the talk today is first I'll spend some time talking in general about microbiota. What do we know in a general sense? And there are some fun facts in there. And then I will look at what type of microorganisms are in our body. And then we'll talk about what is the relationship of the microbiota to diseases, particularly to autoimmune diseases. What do we know about that at this point? And then also finally talk a little bit about how diet can influence microbiome and also affect disease manifestation. Next slide, please. So we'll first talk about the microbiota. What is introduction to microbiota? So many of you may or may not know this, but in our body, we have about 100 trillion microorganisms. And we're talking about microorganisms here. We're not talking about our cells. And so we have a lot of microorganisms living in our body spaces. Whether it's the gut, the skin, the respiratory system, urogenital tract, any sort of the body part that is exposed to the outside environment has a certain number of microorganisms, and the total of that is estimated to be 100 trillion. Next slide, please. So in the general sense, it is thought that the microorganisms actually outnumber the human cells that we have. So the number of microorganisms is at least 10 times more than the number of human cells we have. And researchers have argued about the exact number of this, but in any case, the bottom line is there is a lot of microbes compared to the human cells. And in adult humans, the gut microbiota itself weighs about 2 kilograms. And there are about 1,000 different species that colonize the GI tract. And this we're talking about bacteria mainly, because that is where we know most of the things about. But remember that there could be viruses, there could be protozoans, there could be fungi. And so we don't really know a lot about these other organisms. Next slide, please. So when we think about the genes that are expressed by these microorganisms, there is a total of 3.3 million different genes that are expressed. And this is 100 times more than what we express. So humans express about anywhere between 30 to 40,000 genes, whereas the microorganisms express 3.3 million different genes. And so you can see that we are completely outnumbered by the number of genes that are expressed by the bacteria. And just to give you some basic idea of what I'm talking about in terms of genes, there's a picture down at the bottom, which gives you an idea. So the gene is part of the DNA. So it's composed of DNA, and gene is the one that then translates to what we call as protein. And the protein is the one that finally has some function in the body. And so we're talking about this gene there are about 3.3 million types of them. Next slide, please. So we have known about bacteria that is present in the GI tract for a very long time. This is not anything new. So this is a paper back from 1911. That's more than 100 years back. And it was published in a good peer-reviewed journal showing that intestinal bacteria have gas-forming power, and this power is different between different types of bacteria that are present in the gut. In addition, next slide, please. We also know, depending on the type of the intestinal bacteria that are present, some people can get intestinal diseases and others don't get the intestinal disease. So back in 1911, researchers have been trying to understand how is that some people are getting disease and others aren't, based on what type of bacteria is present in the intestinal tract. So what I'm trying to say is, so we've known about this for a long time. So why is this becoming important now? So next slide, please. So when you look at the papers published on microbiota, so you can go to any database where you can search for papers that have been published on microbiota, you can see that starting from 2010, 2011, 2012, the number of papers published on microbiota is drastically increased. So back in 2018, there are more than 10,000 papers published on microbiota. And so what this tells you is something happened recently that everybody is paying a lot of attention to microbiota. And the real reason for this, next slide, please. The real reason for this is really advancement in technology. So recently, in the last 10 years or so, we have advanced DNA sequencing technology that can sequence bacterial DNA and give us ideas about what type of bacteria are present. The old way of trying to culture bacteria and identify the type of organisms is very time consuming, and it doesn't work all the time. Whereas the DNA sequencing technology is very time efficient and gives us enough data to really understand what type of bacteria are present in the body. And so therefore, National Institute of Health, as well as the European sister agency, both of them put a lot of money into the research in terms of understanding the microbiota. And so it's called the Human Microbiome Project. It was established in 2007. And as you can see, that is one of the reasons now we have an idea about different types of bacteria that are present in the body. And so because of this, next slide, please. We now have an almost complete catalog of intestinal, periodontal, that's in the mouth, and the skin microbial community. So we have an almost complete catalog of what type of bacteria are present in these different regions of the body. And now people are trying to put more effort into trying to also understand what type of bacteria are in the lung, what type of bacteria are in the urogenital tract, and so forth, and trying to really understand what these bacteria or microorganisms are doing in the body. Next slide, please. So based on what we know so far, we know that microbiota affects a lot of different systems. So I'm just showing a snapshot of some of the systems here. We know that microbiota can affect the brain behavior. It can affect the cardiovascular system. It can affect the respiratory system. It can affect the kidney function. Obviously, we know that it also affects the GI tract. It can affect the skin, the immune system, and also the musculoskeletal system. So my lab in particular is trying to understand how microbiota is important for bone health. And so there are several researchers that are working on how microbiota influences all these different organs, both under physiological conditions as well as under pathological conditions and disease conditions. Next slide, please. So that gives you an overview of what microbiota means, what type of microbes are in the body, and so forth. Next, what we'll do is look at the types of microorganisms that are in our body. What type of organisms are in our body? Next slide, please. So before I do that, I just want to give you a basic overview of what kingdom and phyla and classification all these different organisms belong to, because as you will see when I'm talking about bacteria, I will talk a lot about phyla, and you need to know what that means. And so in a general classification, as you can see, there is kingdom Animalia, we belong to the kingdom Animalia. And there's also a plant kingdom, there's also a fungi kingdom, bacterial kingdom, and which then all these things are further divided into further classifications. Next slide, please. So when we talk about gut microbiota, we're talking not only about bacteria, but we're also talking about viruses, fungi, as well as protozoans, and how they are interacting together to mediate some form of beneficial functions, normally speaking. And so, but a lot of data that we have so far, a lot of them are focused on bacteria because of the reasons I told you earlier, which is the DNA sequencing technology for the bacteria is really advanced. And now people are trying to put more money into trying to understand what type of viruses, what type of fungi are there, and how are they able to influence the not only the rest of the microbiota, but also the host functions. Next slide, please. So getting back to that classification, so when we talk about bacteria, we're talking about the kingdom bacteria, and then we are talking about phyla. When we talk about phylum, we're talking about the classification at that level. The actual species is further down, okay? So each kingdom has several phyla. There are several classes in each phyla, several orders in each class, several family members in each order. And so you can see there are several species within a genus, and there are several genera within the family. So you can see that this just balloons out in terms of the number of species that can contain within a particular phyla. So, but a lot of the DNA sequencing technology until recently, it's gotten better now, but until recently, are able to identify bacteria at these levels, at the phylum, class, and order. And the species level is only, people are focusing on that now. Next slide, please. So in the GI tract, there are two major phyla that are important. One is called the Firmicutes, the other is the Bacteroidetes. So these two are the predominant microbial communities in the GI tract. In addition to this, there's also what is called as proteobacteria and actinobacteria. Both of those are also present in the GI tract. And again, I'm just using GI tract as an example. If you take other systems like the respiratory tract or the urogenital tract, the distribution of these phyla or the composition of these phyla will be different. Next slide, please. So when we can look at what happens to microbiota composition, or in other words, in an individual, what happens to these bacteria over their lifetime. So what is shown here in these circles these are color circles. Each color represents a certain phyla of the bacteria. So for example, the pink color represents the Firmicutes, the dark green represents the actinobacteria, and red represents the proteobacteria, so on and so forth. So what this tells you is that when babies are born, we have a certain composition. And that composition is again dependent on whether the baby is breastfed or formula fed. And when the baby switches to a solid food, again, the composition differs. So as you can see here, you see a lot of a red color here, whereas it becomes yellow, which is the other types of bacteria that take over. So clearly, this tells you that diet has an enormous influence on the type of the bacteria that are going to be present. As one ages through, again, the type of things that are important in terms of what the person takes. For example, if the person is on antibiotics, that is going to change the type of the bacteria that are present. If the person is healthy, that is going to be different from a person who has malnutrition. So as you can, based on the color, the types of bacteria can be different in a toddler. And as one ages and becomes an adult, again, it acquires a certain composition. But if the adult has other diseases, for example, obesity, then that can also have a different gut microbiota composition. And as one ages, again, the microbiota composition becomes different. So what is the point of this? The point is that as we go through a life cycle, the type of bacteria that are present in our body also changes depending on what type of things we put in our body, like the antibiotics or the diet, and what type of healthy lifestyle we're leading. Next slide, please. So this slide is, I know it's a very busy slide, so don't focus on a lot of the bacterial names. What I want to sort of convey here is starting from the gestation time in a pregnant woman through adulthood, there are a lot of things that differ. And even within that, when conditions are different, for example, when we think about delivery mode, a C-section versus vaginal delivery, the type of bacteria that are going to colonize in the baby is going to be different. Again, whether it's a preterm or term baby, the type of bacteria that colonize is going to be different. So as you can just quickly glance at the type of bacteria, you can see that there are some similar bacteria, but there are definitely different types of bacteria. And again, whether the baby is breastfed or formula fed, again, those also differ in terms of the bacteria that colonize the GI tract. And again, as I said before, the diet is a major influence. And eventually, as the baby, at about 3 years or 4 years old, the type of bacteria that are present in the GI tract somewhat stabilizes. And in the GI tract, you've got a lot of Firmicutes bacteria and Bacteroidetes bacteria, and also some Proteobacteria and Actinobacteria. So the point of this slide is that over time, starting from birth, depending on what route is taken, the type of bacteria that are going to colonize is going to be slightly different. Next slide, please. So again, this is sort of the color coding for the different types of phyla for the bacteria that are present. And these different types of phyla are present in the different regions of the, sorry, one second, please. These different types of bacteria that are present in the different parts of the GI tract is different. So what I mean by that is when you take, for example, the mouth. So mouth has a certain composition of the bacteria. You see a lot of yellow, which is the Firmicutes, and some of the other colors. But as you go from the mouth through the esophagus and the stomach and the colon, you see that the color changes, meaning that the type of bacteria that are different across the GI tract. And so as you also look in the skin has different types of bacteria, and also the urogenital tract has a different composition of bacteria. So the point of this is, as we go through the different parts of the GI tract and look at the skin and other parts of the body, we see different compositions of the bacteria. Next slide, please. This is, again, not to bother you with too many details in terms of the type of bacteria that are present, but just to give you an idea that as you go from the upper part of the GI tract to the lower part of the GI tract, the number of bacteria that are present also changes. So for example, stomach has about 100 to 1,000 different bacteria, whereas as you go to colon, you have enormous amount of bacteria. There's a 10 to the power 12 numbers of bacteria in the colon. So what this tells you is that the different regions of the GI tract colonize different types of bacteria depending on what conditions are favorable for the bacteria. So in some cases, some bacteria like acidic environment, so they are in the stomach. Some like a slightly alkaline environment, so they are in the small intestine. And so depending on what type of environment they like, they tend to be in different parts of the GI tract.
Doctor, I'd like to
Next slide.
just ask a question. This is John Mahalski. Now, do we see a difference between, as I'm assuming like on this slide, we're showing typical. Now, do we see the differences or know what the differences are in a scleroderma patient as represented as the amount of bacteria that we would see in the stomach, small intestine, or colon?
Unfortunately, no. So this is sort of a general overview of what we are supposed to see. But even within that, even when you take even two healthy patients, healthy people, their composition is going to be slightly different. So certainly in a scleroderma patient or other autoimmune patients, the types of bacteria that are going to be present is going to be different. But we don't know what that difference means at this point.
Okay.
Do you see what I mean?
Thank you. I understand. I'm sure that was a question that everyone was thinking, but I wanted to ask it for them.
Sure. No problem. Next slide, please. So similar to the gut, the skin also has a very diverse and complex microbiota. So the skin microbiota, that together it's called as either the skin microbiota or the skin microbiome. And again, why do we want to understand what type of microorganisms are present in the skin? Because once we understand that, we might be able to make new antimicrobials or promicrobials for therapeutic purposes. We can use these for targeting some of the disease processes that are occurring, because remember, some of the microorganisms are actually good for us. So these actually produce things that are good for our body. And so if we understand what they're producing, then we might be able to actually use that to help some of the patients. Next slide, please. So again, just to give you an overview of the skin, so this is a histology of the skin. So you basically take a skin and make a section and look under the microscope, and this is just a cartoon of it. And when you do that, you can see that the skin surface has a lot of bacteria, fungi, viruses, but also the sweat and the sebaceous glands. These also have a lot of bacteria. So depending on the type of bacteria that are present, that can influence the disease development in some patients. In addition, different people have been, especially in the skin, different people have been shown to have different types of these microorganisms, which is why forensic pathologists are thinking about using this as a means for identifying criminals, because your skin microbiota composition will be different from any other person's skin microbiota. And so each person has a fingerprint of microbiota, and therefore we might be able to tell who was in a location, who wasn't in a location. So this is one of the things that's being talked about quite a bit. Next slide, please. So another more complicated thing about the skin microbiota is that not all parts of the skin are going to have similar microbiota composition. So for example, if you take moist sites versus dry sites in the skin, these are going to have different compositions of bacteria. And so the next slide actually illustrates the complexity even better. So can you go to the next slide, please? So this is a series of graphs. What this is showing you is, again, the colors relate to the bacterial composition. Okay, so if they all have green, they all have the same type of bacteria. So what each of these figures tell you is the HV1, HV2, HV3, and HV4 are 4 different individuals. And the graph that's marked A, B, C, and D are 4 different sites in the body. So when you look at the same site in 4 different people, you can see that the composition of bacteria is different. Also, when you look at the 4 different regions, even for the same person, the composition of bacteria is very different. So you understand the complexity involved in this, because when we are trying to understand how does skin microbiome influence disease, we have to think about all these different things, because we have to be able to understand which part of the skin are we going to look at, which part of the skin are we going to investigate, and is that important for disease processes. And that's why this is very complex. Next slide, please. So having said that, different people have different microbiota, and depending on what you eat and what drugs you take, the microbiota can be different. So the biggest question is, how is the microbiome or microbiota selected, established, and maintained in an individual? How is it possible that so many trillion microbes are actually established in the body? Next slide, please. So the simple answer is we actually don't know for sure. We actually don't know how this is happening. One thing we do know is, next slide, please. The mother's microbiome plays an important role in determining what the children's microbiome is, because regardless of whether it's a vaginal delivery or a C-section delivery, the type of microorganisms that are present in the baby is more similar to the mother. But over time, as I've said before, over time, this also changes depending on the type of nutrition that the baby is going to get, depending on the type of environment that the baby is going to be in. And so, as stated in the next slide please. Diet as well as antibiotics can actually change the type of microorganisms that are present in the body. And so this is why it's very complex we cannot simplify how microbiota influences disease, because we are talking about this complex organ system. And now microbiota itself is called as the organ system. How this complex organ system influences many different aspects of life. And on top of that, different people have different composition of that, makes it even more difficult to understand this. So the next question is there a relationship between microbiota and disease, and particularly in autoimmune disease? So when we think about microbiota and disease, there are two sets of models that we use. One is the animal models that we use for studying, and then we also do clinical trials or clinical studies trying to understand how is microbiota influencing the disease processes. And each of those has certain caveats. So next slide, please. So we know already that autoimmune diseases are really complex and have a number of different genes that are important for the pathogenesis of the disease. And we also now know that genes and the environmental factors are also important. And the environmental factors that we're talking about in this case can be the microbiota. And so a lot of epidemiological studies as well as clinical studies have suggested certain microbial organisms are important for certain inflammatory diseases. But the problem is a lot of these epidemiological studies or clinical studies, causation is hard to demonstrate. And what I mean by that is, just because an organism is going up or down in a patient, that doesn't necessarily mean that the patient's disease is because of that change in organism. In other words, an analogy would be, let's say you wake up every day at sunrise, and that tells you that when you wake up, the sun rises, but that doesn't tell you that the sun rises because you're waking up. Okay, so those two are things that are happening at the same time, but may not be necessarily related. And that is what is hard to prove in human studies. Whereas in using mouse models, we actually have been able to understand a little bit better about how microorganisms can influence influence disease processes. So for this, we try to use what are called as germ-free mice. So we can generate mice without any microbes. So they're clean, they're germ-free. And then we can ask some questions. So what happens to disease processes in those mice? And we can also put one or two or several different known bacteria and ask, so what is the function of these bacteria in these mice? And by doing that, we can take a few steps forward in terms of trying to understand how the microbes are influencing some of the physiological functions of the body. But again, it's a mouse model. Mice aren't human, and therefore we always need to get back to human studies to understand whether some of the changes that we see in the mice are important in the humans as well. Next slide, please. So what we know now is that the role of the oral, gut, as well as the skin microbiome is an important contributor to disease mechanism. What we mean by that is we know that they're important, we don't know exactly how they're important. Okay, we know that when we manipulate some of the microbes in the body, we are able to change the progression of the disease. But exactly how that is happening is really still a big question in the field. Next slide, please. So overall, when we think about microbiota and when we think about health and disease, what we see based on several studies is that a lifestyle that is a healthy lifestyle where you have anti-inflammatory drugs or immune potentiating therapeutics, or a diet that is a high fiber diet, all these things make your microbiota to have a composition where there are more beneficial microbiota compared to harmful microbiota. What I mean by that is, again, let me take a step back and explain this. So when we think about microbiota, in a healthy person, it's predominantly there's more of beneficial bacteria than harmful bacteria. Okay, and so if a diet that is a healthy diet or a healthy lifestyle, what that does is that makes sure that we still have more of the beneficial bacteria compared to harmful bacteria, and we call that as the balanced gut microbiota. And the balanced gut microbiota then leads to a health in terms of overall health. In contrast to that, completely opposite to that, is when people are on Western diets, high fat, high sugar diets, or overnutrition, or sedentary lifestyle, or using a lot of antibiotics. These will lead to more of the harmful bacteria compared to beneficial bacteria. So there's more harmful bacteria in the body compared to beneficial bacteria. And now it leads to what we call as gut microbial dysbiosis, meaning the microbiota is completely out of balance. And that leads to several disease processes. For example, systemic inflammation. It can lead to gut leaking some of the bacterial products that can lead to inflammation in the body, and can lead to, there's also some evidence that that is important for arthritis and some of the autoimmune diseases, also in a lot of other disease processes. Next slide, please. So some people believe that the gut microbiota is sort of the root cause for a lot of different diseases. And even though that's not quite true, gut microbiota certainly influences a lot of these different diseases. So you can see so many branches of diseases here, going from diabetes to diarrhea to colorectal cancer, cardiovascular diseases, hypertension. So in all of these diseases, either in the animal models or in humans, the gut microbiota has been shown to influence the disease process. Next slide, please. So as I said before, In terms of animal studies, we've focused mainly on using mouse models because that's the one that we can make germ-free mice, and therefore we are able to ask some specific questions. And in these mouse models, we are able to show that the microbiota is important in autoimmune diseases. And again, it's easier to show cause and effect in terms of the animal studies using animal models. But in human studies, next slide, please. As I said before, causation is a little bit harder to prove. And so in human clinical studies, even though there are associations or correlations that are shown, many of the studies don't have a causation. And so causation is very hard to prove in human studies. So we take a lot of the data that we get from mouse studies and try to apply that to the humans. Next slide, please. So now we come to the last part of the talk, which is the role of diet and how diet influences the microbiota and how it influences disease manifestations. So there are several parts to this because different people have different diets and different bacteria live based off of different types of diet that you provide to them. And so how do we know which bacteria likes which diet and how that eventually influences our body? And so that's what we're going to look at in the next few slides. Next slide, please. So in terms of how diet influences autoimmune diseases, we've known that for several years back, especially related to IBD patients who have ulcerative colitis or Crohn's disease. The type of diet has been shown to be an important factor in terms of the development of the disease. And furthermore, based on a number of different studies, both in humans and animals, we now know that the nutrition, the microbiota that is present in the gut, the intestinal immune system, and the autoimmune pathology, meaning how the autoimmune disease develops, all these are really intertwined, meaning, for example, if you change the nutrition, that is going to change the type of the intestinal microbiota. And that is going to change the type of immune cells that are going to be present. And that will definitely change the autoimmune pathology. So this is why nutrition on the diet is so important in terms of trying to understand the role of the microbiota in disease. Next slide, please. So this cartoon illustrates what we actually know at this point. The cartoon says if I knew what those trillions of bacteria wanted, I would give it to them. So the problem is we don't know what all those bacteria want. We want all the beneficial bacteria in our body to be able to survive and be happy so that they can produce things that are good for us. But that sort of research is still going on, and we don't know exactly what type of bacteria requires what type of food. But what we do know is that high fiber diet seems to have a better, it seems to be better for microbiota in terms of sort of creating this environment that is better for healing. Next slide, please. So this is a cartoon that compares Mediterranean diet versus Western diet. And so as you can see with these two pyramids, when someone is on Mediterranean diet, where there's a lot of fruits and vegetables, lots of fiber, and very little of fat and meat products, their microbiota is happy. Okay, so microbiota symbiosis, meaning they're happy they're more beneficial microbiota than harmful microbiota. But when somebody is on Western diet, where the majority of the diet consists of high fat, high sugar diet, and very little of the fiber diet, then their microbiota is actually in a state of what is called as dysbiosis, where they're unhappy. So their microbiota has more of harmful bacteria compared to beneficial bacteria. So this is, again, a lot of this is based on studies from animal models, but also by tracking some of the humans in different regions in the world. people are able to tell that the type of microbiota that are present in humans that eat primarily vegetarian food or high fiber, high fruit and vegetable diet have number of beneficial bacteria compared to humans that consume mainly high fat, high sugar diets. Next slide, please. So this is again a cartoon that even though you may want to eat garden salad and high fiber diet, your harmful bacteria in the body may want cheeseburgers and fries. So you have to make the decision for them not to give those things and eat more high fiber, high fruits and vegetables, those type of diets, and that will help maintain a more healthy bacteria, beneficial bacteria in the body compared to the harmful bacteria. Next slide, please. So why do I say high fiber diet is good for us? What is the good thing that comes out of the high fiber diet? So when we eat high fiber diet, then we have a lot of beneficial bacteria, and just by virtue of being present, these beneficial bacteria outnumber pathogenic bacteria. So this is what is called as competition exclusion. By just being more beneficial bacteria being present, they outnumber the pathogenic bacteria, and that's a good thing. Next slide, please. In addition, these beneficial bacteria also produce certain things. One thing that we know for sure that they produce is what is called as short-chain fatty acids. So these are fatty acids of a unique composition, and these short-chain fatty acids are important for mucus production, tissue repair, anti-inflammatory pathways, epithelial integrity. So if we keep the epithelial integrity high, then there's going to be less leakage of bacterial products across the gut. And it's also important for tolerance. So these products that are produced by some of these beneficial bacteria have a lot of important functions in our body. And that's absolutely critical in terms of maintaining a healthy lifestyle. Next slide, please. So in sort of towards the end of the talk, when we talk about diet, we really need to, I know that we all want to understand how we can influence our diet. And one of the things that you commonly see is what are called as probiotics. And you see this in every grocery store. You see yogurt that has different types of probiotics. And what are these? So probiotics are live bacteria. And these are probiotics are active bacterial culture, so they're live. And there's something called as prebiotics, and prebiotics basically are food for these bacteria, these probiotics. So together, they're called as symbiotics. So probiotics are good bacteria, and they're live. Prebiotics is food for these good bacteria. And sometimes you can have them together, and that's called as symbiotics, where you take a combination of pro and prebiotics so that the bacteria that are present utilize these prebiotics and produce things that are good for us, basically. Next slide, please. And as one of the key things to understand about probiotics is, again, there are so many of these. When you go to health shop or any food store, you can see that different types of the yogurt have different types of probiotics, and we don't really know how these different types of probiotics is important for health. And so a lot of the problem with this sort of these probiotics is that because these are available in food stores, these are not FDA regulated, and so but these aren't necessarily harmful, but we don't know if all of them have beneficial effects in the body. And so when we think about taking probiotics for any disease that we have to be really careful in terms of consulting with our physicians and also checking in the research literature, either you with your physician or together, check research literature to see has there been any studies done on these particular probiotics that has shown beneficial effects in this particular condition. And so that's a critical thing that to do before going out and buying a lot of different probiotics. Next slide, please. So in terms of scleroderma patients themselves, there are some studies that are showing that probiotics can be of benefit to a scleroderma patient. So here's one study that looked at looking at the effect of probiotics in terms of GI symptoms, in terms of moderate bloating, to see whether moderate bloating would improve. And it was a pilot study, and they found that, in fact, using the certain probiotics in that paper definitely helped improve some of the symptoms associated with the scleroderma patients. Next slide, please. Similarly, there are also probiotic studies done in other autoimmune disease patients. So in this case, it's a rheumatoid arthritis patients, showing that this particular bacteria, Lactobacillus casei, but they examined whether this supplementation of this is good for the RA patients in terms of suppressing the inflammation. And they found that this particular bacteria supplementation actually did improve the disease activity and inflammatory status. So I'm showing these two studies to give you an idea that, yes, there are studies out there that will tell you certain probiotics are good, but there are also other studies that show that certain probiotics are not necessarily beneficial for a particular patient. So we really need to dig into the literature to see what type of bacteria or particularly probiotics are good for the patients, and depending on what type of conditions you have. So one of the key things to do is talk to your physician to see whether a certain probiotic is actually beneficial for you. Next slide, please. So in the last 15 minutes or so, what I've done is I've tried to give you some overview of what microbiota means, what type of microorganisms are present in the body, and how they're related to disease development, and also how diet can influence these microbes. And what I want to convey is this is an incredibly complex field, and we're still trying to sort out all the different nuances that each of these microorganisms are important for. And therefore, so anytime you think about changing your diet or changing your or taking probiotics and so forth, it's especially if you have scleroderma or other autoimmune disease, it's important to really talk to your physician to make sure that particular probiotic is actually, whether it's good for your disease condition or not. I think that's all I have. Next slide, please. Thank you for listening patiently, and I'm happy to answer any questions you may have.
Well, first of all, let me just say thank you very much, Doctor. I will say your presentation is rather unique from a professional or professors. I love your sense of humor. So from that standpoint, making a subject that some people would say, I don't know anything about, but what makes it what you want to continue to follow is very entertaining. So
thank you.
I'm going to unmute Michael also, and right now is that I'm going to ask anyone within the who's online right now who has a question, raise your hand and I'll unmute you, and well, you can ask the question to the doctor. And the mute button is, I should say, the raise your hand button is on the top of your bar of your screen if you are online. Well, let me do it this way. Let me just try, because we have a lot of people who called in and they don't have that ability. I'm going to unmute everybody, so please, if there's background noise, please tell people to hold on. So let's see if there's any questions within the community here. So here we go. Unmute all. All right, we have people all unmuted. They're all coming online. So the question is there anybody who has a question for the doctor?
Yes, I have a question about gastroparesis.
Okay.
I'm wondering how, as a scleroderma patient, I have gastroparesis as well. And
Okay.
I'm wondering what challenges does that bring with this whole microbiome talk?
That's a difficult question to answer because so as I sort of indicated, microbiota is different in. I'm sorry, hold on a second. Microbiota is different in different conditions, and even within scleroderma patients, depending on what type of scleroderma you have, that can also differ in terms of the microbiota. And so certainly in terms of talking to your physician to see if there's a way to modify your diet or modify your microbiota by taking probiotics or prebiotics, those are certainly good things to think about to talk with your patient. I know I'm not giving you a straight answer because I don't know the answer to your question. I don't know if anybody knows the answer to that question.
Thank you very much.
Sure.
Thank you. Is there any other questions?
Yes. Can you hear me?
Yeah.
Please speak up, Nat. I believe Natalie.
And
Please speak up.
I have a question.
Mm-hmm.
I have duodenitis.
Okay.
So it's inflammation in
Right.
the duodenum.
Right.
And is that affected by, could that be caused by the microbiome or the microbiota being off, and or can it be helped by having the right microbiota?
Sure. So certainly, I don't know if anyone has directly looked at whether microbiota can cause duodenitis, but I wouldn't be surprised if there are studies showing that microbiota plays an important role in this. And so, again, I don't know if anyone has researched this to that extent. And it would certainly, again, be a good thing to talk to your physician to see whether taking some probiotics would be beneficial for this. And certainly, there are some types of probiotics that have been shown to be important for or good for IBD patients. So when we talk about Crohn's disease or ulcerative colitis, the GI tract inflammation diseases, then there are certain probiotics that seem to be good for them. And so certainly trying to work with your physician to find the best probiotic that could be useful for duodenitis would be worth doing that.
Okay, so one that would help with inflammation then.
Correct, to suppress the inflammation, correct.
Okay, all right, thank you.
all right, I'm with you.
Thank you very much. Is there any other questions?
Oh, it's over?
Yeah.
So this is Sharon Fine. You suggest that we talk to our doctors and do some research, say related to probiotics.
Mm-hmm.
There's a lot of diet information,
Right.
there, but every doctor I've ever talked to has no idea about diet. They really don't, other than what they've done for themselves.
Sure.
So how would you suggest we do, where are the best sources to go to for research about probiotics, other than advertising?
That's unfortunate if the physicians are not aware of this. But so one of the places you would go is into the National Institute of Health website. And in the website, you should be able to search for probiotics research. Or if you go to, there are clinical trials that are going on about probiotics. So if you go to a website called clinicaltrials.gov, you can search for different diseases, you can search for different drugs. There you can search for specific probiotics and your condition, whatever condition that may be, and see if there has been any study done using those probiotics. And so by doing that, you might be able to find a probiotic that has worked for a similar condition as yours and then you can talk to hopefully your physician to see if that would be a good thing to do.
Okay, thank you.
Sure.
Hi,
Hi.
Anne Wills here. I just wanted to ask, a lot of scleroderma patients have issues with digesting fibre. So what is recommended for them?
Right. So exactly. So this is why I sort of stress talking to your physician, because different patients are different, right? And so and different autoimmune diseases are different. Some autoimmune diseases, patients don't have issues with fiber diet. Sometimes high fiber diet can cause irritation and so forth. So it's really a lot of times it's personalized approach in terms of the diet is really important. I mean, I have a sister who has had IBD for a long time, and she used to go through different types of diet to see which diet was good for her in terms of sort of keeping the IBD down. And so I think a lot of times that's what you have to do, especially if the physicians are not able to find the best diet.
Thank you.
Sure.
Any other questions? Okay, with that, I will say it's 8 o'clock. I will say thank you, Doctor, for your time tonight, and thank you for everyone who called in. Very educational presentation, and I learned a lot. And I guess the one question I had is that back in your presentation, you mentioned that there was the graph on the number of research papers that have been issued during basically the last 4 to 5, basically last 10 years. Extra has gone exponentially up. Is there an explanation why is now more research is being done into this area?
Because everyone is realizing that microbiota plays a very important role in our body, and so so everyone is trying to understand how these microbiota are able to influence these processes in the body. I think it's a simple fact that technology has improved a lot, and therefore we're able to understand some aspects of how microbiota is important for the body.
Okay, one second. I'm going to have to mute. I'm getting feedback from some people. So let me just mute and I'll unmute you, Doctor. Just a second.
Sure, no problem.
Okay. So with that, I want to say I really appreciate your time today. Michael, you are also unmuted. Is there anything you'd like to add?
Yeah, if we're ready to end the webinar, I do have some closing statements. Are we ready?
Yes, go ahead.
Yes. All right. Staying educated about scleroderma is so important, and we can benefit scleroderma patients. This is the reason that the Michigan chapter strategically has created this web-based educational experience. Being educated can help the scleroderma community become better health care advocates. I hope you will join us on August 8th for the next webinar. We will be hosting Dr. Vivek Nagaraja from Michigan Medicine will presenting on the topic Sclero Men, more than just the 20%. This will be another informative and educational live webinar that will be for males only. Thank you again for sharing your time on this webinar.
Thank you for the opportunity. It was my pleasure.
And thank you, everybody, and have a safe night, and God bless.
Thank you.